But today we are looking at the control of muscle tension, which is a review and picking up on the lab exercises you did last week. So everything we discuss, you've done and you have experience with. Control of muscle tension, or control of muscle force, because tension within the muscle is what generates force that can be measured externally. It's technically tension within the muscle, force that is measured externally. How do you increase force? How do you increase force? Muscles can be on or off, and they can be on to varying degrees, but the individual units, when they're depolarized, the base unit of depolarization has one set force. How do you increase force generated in a fiber or in a muscle? One way is to increase the amount of muscle activated. So part of your muscle might be engaged if you lift a cup off the desk or a pen and start writing. Part of the muscle might be engaged. You don't need a lot of force for that. But if you have a heavier object, you need more force, and so first of all, you can activate more of the muscle. That's one way to increase the force of contraction. The second way is to amplify or strengthen, intensify the muscle that is already activated. And you had an example of this in lab where, without moving the electrodes that were attached to the forearm, but increasing the stimulus that was delivered to the muscle, the force of contraction increased. So you can increase or intensify the fibers that are already activated. So there's two ways to increase the force of contraction: recruit more muscle or amplify the activated muscle. In terms of recruiting muscle, the unit of recruitment you know is the motor unit. And the motor unit is described as a somatic nerve and all of its branches and the fibers that it innervates, the muscle fibers that it connects to and innervates. So the nerve, its branches, and the fibers are what constitute a motor unit. And if a nerve is depolarized, it will deliver that action potential to all of the fibers equally and depolarize all of those fibers. So if the nerve heading to a motor unit is on, that motor unit is on. So if we want to increase the force of contraction, well, we can activate more motor nerves. We can recruit more motor units. All of the motor units, or all of the motor nerves, attach distally or innervate fibers of the same type. And you can see that on the schematic shown here. We have three different motor nerves coming out of the spinal column. Remember, anterior is motor, the ventral side is motor, so these are the motor nerves. Three different colors heading to different muscle fibers, and if the color scheme holds up, I'm going to go ahead and say that the red ones are slow or type 1. These are the ones that have a lot of myoglobin, a lot of mitochondria, that are oxygenated, but that don't make as much force. Type 1, slow oxidative muscle, generally looks quite red. And you can see the red nerves here innervate only the red fibers. And the same is true for what I'll call the fast glycolytic muscles, the really light ones, white muscle fibers, and the fast oxidative glycolytic, which I'm calling a hybrid here, that are both fast, have a high degree of force, and have some oxidative capability. They're a well-rounded muscle fiber. The neurons within a motor unit only supply muscles of the same fiber type. I'm throwing these up here as well. We mentioned briefly last class, type 1 is slow, type 2 is fast, and then there are two flavors of fast. Type 2X is the very fast glycolytic only, and type 2A is the hybrid model. So these are the different types of muscle fibers, or at least how we categorize them. So the nerves of a motor unit innervate fibers of the same type, and they're usually distributed throughout the muscle. It looks like they're pretty close together here because you can only show a small bundle of fibers, but they might be spread widely throughout the muscle, depending on the branching of that nerve. They're not always adjacent. So they can be in different areas of the muscle, but they're all the same type. And so if we turn one motor unit on, we get a certain amount of force or contraction. It's generally weak. If we activate more motor units, more tension means more force or a stronger muscle contraction. Activating more motor units makes a stronger contraction. Now, this word many means a couple different things. Activating many motor units results in a stronger muscle contraction. And here it implies that in addition to the slow motor unit, you're going to start to activate the fast or the hybrid motor units. That's what many might mean, at least according to this diagram. And that does happen. Depending on the resistance, depending on the load, that does happen. But it also refers to, or can refer to, activating more type 1 motor units. You can activate more motor units of the same fiber type and still get a larger or stronger contraction. So you heard in lab as well, you can see 3 different fibers per motor unit here, depending on the muscle. If it's a fine muscle that's involved in articulations, like muscles of the vocal cords or the larynx, maybe there's 50 fibers in a motor unit. Biceps, latissimus, gluteus maximus, 1,000 to 3,000 fibers per motor unit. Very dispersed terminal end fibers, many fibers that can be included in that motor unit. Now I want to point out while we're here, because I bring it up on the next slide, the colors are generally what we would expect. The nerves innervate the appropriate muscles, but the diameters aren't drawn to scale. And that's important when we get to the next slide. The red ones are the smallest, and the fast, type 2X, are the largest, and hybrids are in the middle. Diameter is different for the fibers in the muscle, depending on the type. And that's important for understanding the process of motor unit recruitment, or the hierarchy of motor unit recruitment. So this is a figure that you'll have in your textbook, which is a bit more muddled and only has 2 fiber types shown here, and the colors are kind of garish and don't really match up. But the slows are the darker fibers with the green nerves, and the fast, we don't know what kind of fast fibers. They're the lighter fibers with the purple nerves. 2 different motor units shown here. And these could be recruited, one of them might be recruited, or both. Understanding the hierarchy of recruitment is important. Hierarchy is as such. It's according to something called Henneman's size principle. When you're recruiting motor units, when you're turning on a muscle, small fibers activate first. Always. Small fibers depolarize, are recruited first. That means type 1 muscle fibers always activated. The smallest fibers are the type 1 slow oxidative. They're always activated first. If the work that you're doing is too much for those slow oxidative fibers, then you will progressively recruit more motor units. More slow oxidative motor units, and if you need to, larger motor units. You'll recruit the fast oxidative glycolytic, the type 2As, the hybrids. And then if that's not enough, you'll recruit the fast glycolytics, the very strong, very large type 2X fibers. And it always works in this hierarchy. From small to large, from weak to strong, depending on the load against which they are working. Henneman's size principle. That's a law of muscle recruitment, or a law of physiology. Which has certain implications. If you think about weight training or bodybuilding, it helps to clarify or understand why lifting heavy is probably one of the best roads to develop large muscle fibers and large muscle size. The biggest fibers are only activated when you lift against a large load or a heavy load. It doesn't help to understand the pattern of recruitment for endurance-type exercise, cycling, jogging, running. It's not a high-force activity. You don't need to generate a lot of strength or a lot of contraction. Here, that type of exercise is characterized by rhythmic recruiting of slow fibers. Rhythmic recruiting of slow fibers. And so you might turn on and off the same slow motor unit. But over time, even those will get tired and fatigue and not be able to contract. And so what you might find is that other slow motor units, other slow oxidative units will alternately contract to give the first ones a rest, to allow them to recover. So similar motor units of the same fiber type will contract alternately to sustain contractions over longer periods. You won't have type 2X fibers taking over for slow oxidative if you're out for a run later today. That won't happen. So we can increase the amount of muscle that's activated. More slow motor units, or we add fast oxidative glycolytic and fast glycolytic on top, if the force requires it. More of the muscles turned on. One way to increase force. The second way is to increase the intensity of those units that are already active. And so the base unit of contraction in a sarcomere is shown here. It's the twitch contraction. If the muscle depolarizes, you will register a certain amount of force. And this was the very first example in lab that you did with one pulse at a small or medium threshold. You registered some small amount of force. A twitch contraction. So you depolarize the motor unit nerve once. All the downstream fibers also depolarize. They generate some amount of force, and then they relax. And this is what it might look like. We have a few characteristics to look at. There are a few different phases. The latent period, or the latent phase, describes transmission of the action potential. There's no contraction happening yet. The signal has arrived, and it's being transmitted to the muscle, but right now, it's washing over the plasma membrane. It's washing over the sarcolemma and down the transverse tubules. It's activating the sarcoplasmic reticulum to release calcium. But nowhere in there has the muscle started to contract yet. There's a small delay between the signal arriving and contraction happening. That's the latent period. It's exaggerated here. It's a very quick short-lived latent period. Main phase of contraction, the contraction period, is what is described by the actual myosin cycle. Calcium has moved troponin and tropomyosin out of the way. Myosin can create a cross bridge. It undergoes the power stroke, and the cycle continues as long as calcium is present. The muscle contracts. Signal's been delivered, cross bridge forms, force or tension is generated. That's the contraction period. The contraction period will continue as long as calcium is present, but in a twitch contraction where one action potential is delivered, calcium is pulled back out of the cytoplasm very quickly. It's sequestered, it's hidden away. Which is important. It's such a potent signal for turning on contraction in the muscle, for turning on metabolism that we'll talk about later today and tomorrow. It's such a potent signal, you don't want it hanging around. So we pull calcium back in very, very quickly. And when we do that, the muscle can no longer contract. Troponin blocks the myosin binding site. No cross bridges can form. Tension gradually abates, gradually wears off as more cross bridges disengage. That's the relaxation period. What's not shown here, but is mentioned in the book, is the refractory period. And all muscle has a refractory period. Even skeletal muscle, but it's not really important in skeletal muscle. A refractory period is however much time after the action potential is delivered that the muscle is unresponsive to another incoming action potential. We saw that with nerves already. In muscle, maybe 1 millisecond. And I'm exaggerating it for you here. The action potential is delivered, it washes across the membrane, it's inactivated for some small amount of time. 1-2 milliseconds in muscle only. This shows 5 milliseconds. I've exaggerated it here so you can see it. Not really that important in muscle, very important in the heart. Very important in the heart. It's one of the ways that we ensure regular, ordered, and directional pumping of blood in the heart. In the heart, 200, 300 milliseconds. Those cells are inactive for a long time. We'll see that next semester, why that's important. In the muscle, very short-lived, very transient refractory period. The muscle's inactivated for 1 millisecond, but it can receive other action potentials anytime after this period is done, which again, you saw in lab. That is how we increase the intensity of contraction. How we intensify the contraction of already engaged fibers. We send more signals. What I'm describing is called wave summation. When a second action potential arrives before the muscle contraction has fully relaxed, the whole process starts again, but from a new, higher baseline. From a higher baseline force that allows force to increase more than it would with one single twitch, which is shown here in the diagram. Importantly, this signal has to be sent after the refractory period is done. If it's sent during the refractory period, muscle is unresponsive. Luckily in muscle, the refractory period is 1 millisecond, so we don't really care. So it results in a stronger contraction. If I send a second action potential here before the first has worn off, which is what the black line here is supposed to show, a normal single twitch, we see the new higher baseline starting force, and the contraction is stronger. We generate more tension, we generate more force. And if we do this a lot, like you did in lab, multiple pulses sent before the preceding one can fully relax, we get this progressive building of force. This is wave summation. It's wavy. And the force is added together. The force of multiple individual twitches are added together. You know this style of contraction is unfused tetanus. What that means is we allow some relaxation to occur. Tetanus describes completely tense, contracted, rigid muscle. That's not the case here. We allow some relaxation before stimulating it again. But if we send those action potentials faster, there's no time to relax. If we send the action potential during the contraction phase, there's no time for the muscle or the fiber to relax, and we get this progressive building of force within the fiber. This is how we intensify the units that are already contracting. So what would you guess the mechanism is? If the muscle's depolarized, it should make force no matter what. If I depolarize it twice, why doesn't it just make the same force twice? Why is it additive? And maybe you talked about this in lab. In the lab that I was in, no one knew the answer. Does anyone remember? What allows force to build? What's responsible for contraction? It's a dangerous time to adjust your hair. It looks like a hand raise. You want to give it a shot? Is it what, sorry? It would have resulted in one of the graphs you did in lab, yeah. Why does the force go up? Okay, multiple motor unit summation. I like that. So you might be recruiting more motor units, and that's absolutely possible. I'm going to limit our discussion to one motor unit. If we intensify that single one that's active, why is it able to make more force? Temporal summation, yes. We activate it more frequently. But what is the mechanism? When you activate it more frequently, what happens in the muscle to make it generate more force? What's the signal to initiate contraction? Don't tell me that you don't know that. The signal that initiates contraction, that allows the cross bridges to form. The feed forward early warning system. Yes, absolutely. Calcium. You have some amount of calcium released that immediately gets pulled back in. If you send a second action potential before the muscle is fully relaxed, before calcium has fully been sequestered and pulled away, more calcium is released, there's more available, more cross bridges form. More and more and more and more calcium is released. That's why force can increase. Our final is 2 weeks away. I'm just going to put that out there. Lost the study between now and then. So more and more and more and more calcium allows more cross bridges, allows more myosin binding, more power strokes, more force. To a point. So I'm showing you here the difference in the duration of contraction and the amplitude, which is the force. The size of these boxes is progressively larger, And noticed in the fused tetanus example, it plateaus. What would the plateau mean? If I just told you force builds because more calcium is released and more myosin can create cross bridges, why would it ever plateau? Nice job. You only have so many cross bridges. You only have so many myosin molecules that can make cross bridges and pull on actin. At some point, it's saturated. They're all engaged. And that's what characterizes this plateau. Okay. That's better. So you consciously engage muscle to generate force. When you lift up your computer, lift up a dumbbell, you're consciously engaging muscle. Muscle is also unconsciously engaged. And we describe that as tone. There's always some tone within the muscle, some tension. And usually that accomplishes a goal. You're never fully relaxed. You feel that now more than ever with 2 weeks left in the semester. You're never fully relaxed. There's some resting tension called tone within the muscle. And going back to the idea of alternately contracting motor units, tone is maintained by periodically activating the different slow motor units within the muscle. It's not always one, it would eventually fatigue. There's periodic activation of the slow motor units within a muscle. And this ensures not that the muscle shortens or that the muscle contracts, but that it's firm. It's not flabby or flaccid or loose, it's firm, it has tone, there's tension between the tendons. And its goal is mostly postural. You sitting up straight in your seats, you don't notice it, you're not actively engaging the muscles of your lower back. But there is some basal tone which allows you to sit upright. If you completely relaxed, you fall back and your head would hit the person behind you, or you'd fall over your computer. There's some basal tone that allows you to stay upright. There's some basal tone that allows you to keep writing. That allows me to stay standing vertically, and I don't have to think about it. And that keeps us warm. A lot of the basal tone has to do with generating body heat. You're always using ATP, you're always contracting in some small amount, and the muscles are generating heat. And just think about if you're not hot enough, if you get too cold, what happens then? Shiver. It's an accentuated basal tone. The muscles are contracting, you're not moving with the purpose of generating heat. So there's some basal tone within the muscle that you don't have to think about. The types of contraction that you think about are isotonic and isometric contractions. But I really don't like the word isotonic. Isotonic and isometric contractions. Isotonic is what you think about moving around at the gym. If there is a change in the length of the muscle, a change in the length of the sarcomere, we call that isotonic contraction. And the definition, iso means stable or the same. Tonic means tone. So literally, tone is the same during these contractions. Tone is the same. So maybe you're getting a sense of why I don't like the name of this type of contraction. Tone's not the same, is it? Isotonic, tone is the same. If the muscle shortens, we call that concentric isotonic contraction. If the muscle lengthens, we call that eccentric isotonic contraction. We just spent time last class talking about why this word is not appropriate. Tension's not the same. Tension's, it peaks in the middle and it's lowest on the extremes. Tension is not the same through contraction. As the muscle shortens, it changes. So it's not isotonic. This is a misnomer. But you get enough people together and they use the same term frequently and it sticks. So it's called isotonic when there's a change in muscle length, even though the tone, the tension varies. If there is no change in muscle length, we call it isometric. Iso, stable or the same. Metric, unit of measure. The measurement of the muscle is the same. And you can consciously do both of these. You can sit there and clench your arms, clench your biceps and triceps, and there's no movement. That's an isometric contraction. If you close the angle at the elbow, that's a concentric contraction. If it opens, it's eccentric. And all that means is the force that you're applying is greater than the resistance if it's closing. And the force you're applying is less than the resistance if your arm is opening. We see that illustrated here, picking up a heavy book, probably referring to your anatomy textbook. Concentric contraction on the far left. You can generate force that is greater than the resistance provided by the textbook. It weighs a lot, but your muscles are stronger, and so the joint angle closes. And if you relax and slowly lower, you're producing force still, but it's less than the resistance provided by the book. And the joint angle opens. If you're holding the book level, no movement, isometric contraction. So muscle can be active in each of these scenarios. It's not basal. You can consciously do it and achieve each of these three examples. It's weird to think about contracting the muscle and the muscle elongating, right? Myosin only works in one direction. Myosin only activates during the power stroke. The head only cocks in one direction. It only crawls one way. And so the theory, you've probably heard that eccentric contractions cause a lot of muscle damage. If you ever go to the gym and you do really heavy lifts and you're doing eccentric muscle contractions, it causes a lot of muscle damage. It's because myosin is pulling in the opposite way that the resistance is, and the resistance wins. So myosin's trying to cock its head and walk up actin, but it's essentially pulling the stalks apart. All the tails of myosin that wind together and make the filament, they're being pulled apart as the muscle elongates while under load during an eccentric contraction. But I digress. You'll get more of that in later years. Isotonic versus isometric shown here. And isotonic's probably not the best way to describe shortening or elongating of the muscle. Let's introduce metabolism. I kind of want to leave this till tomorrow, but I also love this topic, so I'm going to preface it a little bit. And it's going to be an easy, light preface. Incidentally, this is a basic map of metabolism within the cell, within the muscle. All of this up here. It's more complicated than this. And you'll learn this, hopefully in later years, not here. But you've probably heard of some things like the citric acid cycle. We've got glycolysis in the middle, fatty acid biosynthesis, protein breakdown, making nucleotides, The mitochondria are way down here. There's oxidative phosphorylation. Lots of stuff going on. Very complicated. We're going to talk about a few of these. Why do we even need to talk about metabolism? Doesn't the muscle just contract? It does. Muscle contracts, and we know that it uses ATP, the energy from ATP, shown here, to contract. There's a lot of energy stored between these phosphates. When one breaks off, that energy goes somewhere. The muscle takes it, does work. Metabolism, you might have some concept of what that is. It describes all of the processes, all of the chemical processes in the cell. So all the processes on the last slide make up metabolism of the cell. It's not good or bad. You can't have a fast or slow metabolism. You can change your metabolic rate by exercising or resting. But metabolism is made up of two parts. The yin and the yang, the white and the black, the good and evil. Catabolism and anabolism. Anabolism is what you think of when you want to grow or build tissue. It's a constructive set of metabolic processes where you make something. You make more complex molecules. Like muscle protein, anabolic steroids, anabolic proteins, or anabolic processes build proteins. Catabolic processes, on the other hand, are destructive or degrading processes where those complex molecules are broken down. A lot of what we're focusing on are catabolic processes. Why? Why do we need to consider metabolism? Simply put, muscles use ATP. ATP needs to be replenished. So, all of the systems we're going to talk about, their ultimate goal is make ATP. Muscles using it at a very high rate. All of these are supports. All of the metabolism that we're going to talk about are supports that make ATP for the muscle to use. So how do muscles make ATP? Myosin doesn't do it. Actin doesn't do it. None of the contractile proteins do it. Completely different systems are responsible for making ATP. And there are two general classes. One is most correctly called substrate level phosphorylation. So a molecule is directly phosphorylated. Substrate. The molecule, the subject of the reaction, is phosphorylated. And we're going to discuss at length creatine phosphate and what I'm putting in quotes as anaerobic glycolysis. I'll tell you why I think that's wrong. And a second class, oxidative phosphorylation, aerobic metabolism. All of this occurs in the mitochondria, and you've heard to it referred as cellular respiration. These are the two classes of metabolic processes that make ATP, and we'll dive into those in depth tomorrow.